Hello everyone, we are Group 5 and our topic is the importance of targeted next-generation sequencing using in cytogenically normal myeloid malignancies. And this paper is published on International Journal of Equality in Health and at 2021 January 19. And the reason we choose this topic is because there are lots of usage about NGS in genetically, and we want to know more about how the NGS usage in the genetics, so we choose this paper. And in this presentation, we will short introduction to this paper and tell about their materials and method and their results and conclusion. And we will have some discussion and our questions after let everyone know this paper.
First, I'll start with the introduction. Myeloid malignancies originate from hematopoietic progenitor cells and are characterized by defective differentiation of myeloid progenitor cells. Advanced molecular detection techniques have changed the diagnostic algorithm of cancer. The increased NGS usage can help change the scope, timing, and suitability of genetic testing in hematopoietic malignancies. Recently, the diagnostic algorithms for AML, NDS, and MPN have evolved due to advances in NGS. The increased knowledge of genetic abnormalities have led to a reclassification of AML. The WHO and European Leukemia Net added new subgroups of diseases, and molecular genetic abnormalities have also been added in diagnostic criteria. An increased number of mutational, epigenetic, and expression studies will help identify the novel markers in myeloid malignancies. NCCN ELN guidelines and some studies have suggested adding some mutation genes to the risk assessment. NGS-based myeloid gene panels will help for the identification of multiple recurrent somatic mutations in many AML patients, and additional molecular genetic mutations can be detected in most cases, even within defined AML entities. In MDS, NGS allow detecting molecular mutations in approximately 90% of patients. As a result, NGS data should be interpreted alongside other laboratory findings, such as cytomorphology, histopathology, immunophenotyping, conventional molecular genetics, cytogenetics, and clinical diagnostics. Now, let me go through the material and methods. First is patient samples. This study included 100 patients from December 2017 to August 2020, and the median age was 54. Their DNA was isolated from bone marrow and peripheral blood and was quantified by cubic fluorometer. And the table below is the WHO classification of their cohort and the result of genetic analysis. One of the methods in the study is next-generation sequencing. For evaluating myeloid neoplasm-specific 141 genes, they used the Human Myeloid Neoplasm QIAsec targeted DNA panel. This panel covers exon-intron boundaries, shown in the figure next page. This is the mechanism of unique molecular indices. They wanted to know if this study a PCR or sequencing error or a true low-frequency mutation. They use the type of barcoding called unique molecular indices (UMIs) before amplification. After amplification, if there was only one start site among the 5 sequences, it was a false variant. However, if all of the sequences contain the start site, it is a true variant. And this table lists covered genes and related diseases in the NGS panel. Next, myeloid sequencer was used for sequencing of amplified targets according to the manufacturer's protocol for paired-end sequencing. Data analysis and quality assessment for coding of single nucleotide variants and analysis of short insertion and deletions were evaluated using Ingenuity Variant Analysis (IVA) program. Amplicon with coverage below 100 times were excluded, and variants with an allele frequency over 5% were investigated further. Libraries covering the target genes were prepared according to the QIAseq target's DNA panel protocol. Following the target enrichment process, libraries were sequenced on the myeloid system and NextSeq 550 system. OCI analysis was used for quality control and variant code format file generation. Variant analysis has been performed in Ingenuity software. Variants were interpreted according to ACMG 2015 recommended standards. The candidate variants were annotated by ENNOVAR with SIFT, polyphen2, mutation test2, and EXAC and other databases. No hotspots or clinically significant variants below standard threshold were confirmed with orthogonal methods such as single sequencing. They also performed a cytogenetic assessment. The first one was karyotyping. They used marrow max and trial media to culture bone marrow and peripheral blood specimens, and incubated for 24, 48, 72 hours in a carbon dioxide incubator, and used cosmic to arrest metaphase cells and stem chromosomes slide by G bending protocol. They used ISCN 2016 for reporting and analyzed 25 metaphases in each culture. The best metaphases were chosen for karyotype analysis, and the total chromosome count was usually determined in 25 cells. The second one is fluorescence in situ hybridization (FISH). They analyzed 200 interphase cells for each sample, and captured images by applied imaging or cytovision system. The final results were reported by using the laboratory-established cutoff for each of the tested probes. Specific gene panel for FISH were applied for each malignancies. FISH panel for AML and MDS are listed below.
I'm going to talk about the result of this paper. They do the next generation sequencing on 100 patients. NGS of hotspot regions of 141 genes has been performed in 100 bone marrow samples referred from the Department of Hematology. Variables with a depth of coverage above 100 times and an allele frequency of above 5% were included in this study. According to the gene and clinical effects, variants were classified as pathogenic and possible pathogenic. 2 or more pathogenic variations were identified in 61 out of 100 patients, and a total of 24 novel pathogenic or likely pathogenic variations have been described. Their novel variations identify and distributions of frequent mutations in AML and NDS. In AML, novel pathogenic and likely pathogenic variations were identified in EP300, STAG2, CUX1, U2AF1, RUNX1, GNAS, CHEK2, CREBBP, and PHF6 genes. In NDS, novel pathogenic and likely pathogenic variations were identified in SRSF2, ASXL1, ATN, RUNX1, and TET2 genes. Next, the distribution of frequent mutations in AML includes TET2, TP53, FLT3, and IDH2 genes, and the common pathogenic variation in NDS was SRSF2. These results show that NGS pathogenic variation detection rate varies in disease groups. In AML was 85%, in NDS was 23%. Likely pathogenic variation detection rate and loss detection rate of NGS have been listed in Table 2, and the mutation list of disease groups has been shown in Table 3. There are Table 2 and part of Table 3 in the paper. In Table 3, they list all of the identified mutations and their distributions of disease, and also label the novel mutation.
And let's talk about the discussion and question about this paper. So, we can know that the NGS in hematologic malignancies can perform a considerably faster and less expensive test compared to other methods like FISH or cytogenic and RT-PCR. Okay, and the NGS usage in leukemia can be used to clonal IgH and TCR rearrangements in MRD, and also it can identify mutants or clonal DNA in several circulating tumor cells, and also it can discover new mutations in the neoplasm, enable us to have the more understand about the prognosis and pathogenesis. And with the mutants identified, we can find out the growth chromosome like the translocated and dislocated and ejected, etc. And the research of interest by other researchers compared to this paper. So the research before about the Northrop, they gave the variance is 53%, and the Kawata note the abnormal NGS is 32.8%. And the other results finds out that the detection rate was 61 higher than 8% about the Northrop, and the detection rate of the NDS was 23 was less. So the difference between them is because the patients have the abnormal cytogenic reports were excluded in this paper, so the mutant frequencies was lower. And to the other, the U have the find out the mutation frequencies was 85.8 in AML cases, but in this paper, the detection rate of NGS in AML was 85%. So they are most similar confirms the importance of NGS testing as a diagnosis tool. Okay, and they also find out the suggest algorithm that prevent the false positive rate. So they have the 100 new diagnosed malignancies, and they will do the karyotype analysis and also the FISH analysis. And after that, they will do the NGS based on 141 gene panels, and it can identify lots of things. So it can prevent the false negative results. And in the summary, the NGS-based testing is widely accepted in the clinical practice, and also the target NGS panels can evaluate the genome-wide numerical imbalance. Also, the NGS testing cannot identify structure abnormalities and lacks single cells and a low target density, but I think the NGS can find out lots of things about a disease to let people to do a diagnosis. Okay, and the question we want to ask because it's about the precise quantity that the NGS is faster and cheaper than testing like FISH and RT-PCR. So in the paper, they just talk about it is more faster and cheaper, so we can use it, but we cannot know. We cannot know like how cheaper, like less than 100 USD or just 10 USD dollars. And the second question is about we want to ask, can the false positive result also have the algorithm to prevent? As in the paper, it just talk about the false negative result, but not about the dealing with false positive results. So we want to know that it also have a algorithm to prevent it and make a better results. Okay, and this is this paper's reference. And this is our work distribution charts. Okay, and this is the end of our presentation. Thank you.